Professor Hébert, or Terry, as he prefers to be called, is a professor of pharmacology and therapeutics at McGill University. He's joining us from Canada, and his focus are GPCRs. So today we will hear a lot of about GPCR signaling in this session as well. And in this case, we will focus on iPSC-derived cardiomyocytes and the GPCR-based signaling in those specific cell types. Terry, thank you very much for being today with us. And the floor is yours.
Thank you very much. I really wanted to be in Munich rather than my extensive teaching schedule here at McGill, but I'm happy to be with you at least virtually. So, and I'd like to thank Elena and Nana for inviting me. I've really enjoyed my interactions with them over the years, and I think we've used their technology extensively in the last 2 years anyway. So some of that I'll show today. So I guess the problem I'm trying to solve, which is weird because I'm not a doctor, is how do we make medicine less one size fits all? And can we personalize medicine? And can we sort of start to imagine a future where we have some tools to build personalized disease models and generate an iterative bench to bedside sort of set of interactions that would allow us to kind of personalize or at least cluster medicine a bit better. So here's the problem, I guess, in two of our patient clusters. So the HID moniker stands for Heart in a Dish. That's the name of our project, and the Heart in a Dish project is based on studying patients with dilated cardiomyopathy. So these are two patients who actually developed dilated cardiomyopathy after being treated with doxorubicin for breast cancer. They're both women. One actually ended up recovering and never needed any hospitalization or no advanced therapies. Both were treated with the standard cocktail of beta blockers, ARNIs to inhibit the angiotensin receptor system, mineralocorticoid antagonists. The second patient actually needed additional medications to manage the disease and proceeded to get a defibrillator implanted and ultimately had a heart transplant. So how do we distinguish between these two patients in terms of predicting who will need advanced therapy, predicting who would recover, rather than just giving them the same meds and hoping for the best, right? Which is essentially what we do now. So how can we personalize treatment, moving away from the current standard of guideline-directed medical therapy, this GMT, where patients get all of these drugs every time they're diagnosed with heart failure, no matter what the cause, no matter what the sex of the patient, the age of the patient, et cetera, et cetera. And then we add on some adjunctive therapies. We proceed to devices, and in some cases, we proceed to more advanced therapies, right? So including transplants. The therapy starts with finding the highest tolerated dose for each patient, and in that sense, it's really one size fits all. So how do we move away from that? And for me, this is a kind of a paper that changed everything, right? This is a paper from Joe Wu's lab, where he actually showed that following the discovery that Shinji Yamanaka made in being able to make stem cells from somatic cells or from adult cells, he was actually the first here to make cardiac myocytes from patients with dilated cardiomyopathy. And he was able to show that the myocytes preserve features of the disease, especially where there were mutations that were driving the disease. And he was able to show that treatments that affected the patients also affected the outcomes for the cell models, right? So it kind of transformed the ability to do disease in a dish modeling and moved us away from animal models in a sense that to study animal models or to use animal models, it's difficult because the myocytes are quite nice, quite lovely when we isolate them from rats or mice, but they only last 2 days in a dish. So it's impossible to kind of track disease progression over time. It's impossible to look at therapeutic regression, et cetera, et cetera. And the question I've become very interested in is often associated with this subclinical progression of disease, right? I know from my own life, now that I'm an old man who's getting medications for hypertension and whatnot, that I refused to go to a doctor until I had symptoms, right? Until my wife noticed things and made me go. But can we model the disease before that happens? Can we intervene before that happens? Can we make my wife less unhappy about the way I take care of myself? And I think human induced pluripotent stem cells offer a way not only to do that, but in a way that can combine it with all of the technology that has developed to do drug screening in such cells, right? So it really is a great combination of the technology associated with screening and the technology associated with being able to make any type of cell in the body from a fibroblast or a blood cell. So I won't talk too much about our patients, but we have a pretty large patient population, almost 2,000 patients in the clinic, and almost 400 of them have agreed to donate blood to have stem cells made. And we're into this now. We've got about 50 cell lines that we've made, and we're trying to get healthy control subjects as well. They have different etiologies. The thing that we like most right now is that the sex of our patients is about equal. They're all mostly getting directed medical therapy or have had it, and some have recovered, and some have not. Right, so it's a really wide swath of the population. So if we could model these things, model the disease in cells made from these patients, it would be pretty interesting, I think. So as Elena introduced me, she told you I'm a GPCR person. It's been a hard transition for me to go away from being a GPCR person, so I don't. But now instead of using the HEK293 cells as my base model, we're using the induced pluripotent stem cells from control subjects and patients. But we're interested in the same receptors, right? The same receptors that are involved in the day-to-day functioning of the heart, so the beta-adrenergic receptors. Also, the ones that are involved in the more sort of kind of healthy life of the heart, right? Like that start to show different signaling patterns when hypertension is sensed, or when there's a damage caused by a myocardial infarct or a heart attack. And the signaling pathways that we can track in those cells, we can track pretty carefully downstream of many different receptors. And we can watch, if you like, those signaling pathways evolve or change during the course of the disease or between control subjects and different patients. So we're interested in tracking the signaling downstream of these four receptors to start. We've gotten into some others we won't talk about today, but and we're very interested in trying to adapt our technological approaches to using these cells. So one thing that we've also started to do since the pandemic, we were trapped in our homes. We thought, what tools do we have that we could kind of develop during that time? And one of the things we started to work on was to build a single cell FRET pipeline in the lab, develop the tools, the code, and all of that to kind of move away from the bulk analysis of all the cells in a dish to segregating populations, right down to the single level into their variations in response. And so we're mainly focused on two endpoints, ERK signaling and pKa signaling, either in the nucleus or in the cytosol. The one I'll talk about today mostly is looking at signaling downstream of pKa. But the idea here is now that instead of getting a bulk response, you can actually see every single cell on that dish reporting. You have some cells that give you strong and stable responses. You have some cells that don't respond. You have some cells that give intermediate responses. And as you'll see, you have some that give more transient responses a bit later. So it's really a powerful tool that we've come to use to interrogate pKa signaling. pKa is important, right? Because it's what allows the heart to manage its beating rate, right? When you stimulate with the beta adrenergic receptors, you activate pKa. pKa can increase the activity of the L-type calcium channels, so more calcium in. pKa activates the ryanodine receptor, so more calcium released, more contractility. But at the same time, pKa also phosphorylates SERCA, which activates that and allows the cycle to end by pumping the calcium back into this sarcoplasmic reticulum. So it allows the heart to beat, but then it also facilitates relaxation so the heart can beat again. So it's a pretty important pathway. And so we've built two forms of the biosensor, cloned by phone, two different forms, and put them into the vectors we like. They're based on enhanced GFPs, and one is localized to the cytosol, and one is localized to the nucleus. When PKA is phosphorylated, when PKA is activated, the sensor now becomes activated as well. And so we can actually see that in the iPSCs now, where you can see that vehicle does nothing. Isoproterenol, the standard beta receptor agonist, actually stratifies. So you have some very weak responders, you have some strong but transient responders, and you have some very strong and stable responders. And that's true in different numbers for different agonists for the receptor, right? So we can kind of find those different populations. Maybe it's not going to be so important to understand the responses in a cell type alone on a dish, but I'll tell you why we're interested in the clusters as we go forward. All right, so that was a control subject. So we started to compare like patients to controls. So these are age-matched female control subject and a patient with dilated cardiomyopathy. It looks pretty much the same across the board. Lots of things activate PKA. Of course, the beta receptor agonists are the strongest activators of it. You would not expect in the alpha-1b to activate the GS pathway or the PKA pathway, and it doesn't in the control, but it's clearly upregulated in this patient, right? So that was a bit of a surprise to us, and that's I guess it shouldn't have been a surprise because we've published several papers that showed that the alpha-1b could couple to GS, right? So here in HEC cells, back in the old days, we would actually look at an EPAC sensor for cyclic AMP, and you could see that there's a nice response to phenylephrine, and it's lost when you knock out GS, and it's restored when you return GS to those cells, right? So, and that was true for the alpha-1A or the alpha-1B. And we could also see that when we started to look in cardiac myocytes. So this was in neonatal rat cardiac myocytes just before the stem cell era started in my lab. So interesting. The alpha-1B seems to be almost like an inotropic reserve system, okay? Now, the first paper I ever published as an independent lab, I guess I should have read that in the intervening years because it looked like when we overexpressed the alpha-1b in a mouse heart, we got a dilated cardiomyopathy. So that tracks very well with what we're seeing now, right? We're seeing that one patient, at least for now, shows an upregulated alpha-1 signaling that may actually impact the course of the disease. So we're interested in that. And so we did the same thing with a male control subject and a male patient, and we didn't see anything related to alpha-1b upregulation, right? So that, again, suggests that the power of the approach is being able to determine patient-level differences, or at least sex-level differences between patients and controls. What we did see in this case was that the tone, the ability to activate PKA is markedly lower in the patient. So there's some loss of contractility that's probably associated with that, right? So that's where we are. The other biosensor that we spend a lot of time developing and using, again, cloning by phone and putting into the vectors we like to use, is this calcium biosensor. So it's a genetically encoded calcium sensor that is localized to the myofilament. So it really only lights up in cardiac myocytes, which is kind of cool for what I'm going to tell you about later. And we can do that same type of experiment where we look at pre and post ISO, and you can see that every time the cells flash, that's when calcium is being released from the SR, and it tracks with contractile. And it's also sensitive to drugs in the sense that when we stimulate with ISO, the cells get brighter, and the frequency of the beats is increased. Won't show the other one. So we also see differences now when we compare the control subject with the DCM patients, and it's pretty clear that there are like the gray are the control subjects. There's only five people here, and the red is the patients. And there's differences that we can actually track, right? So there does seem to be compromised calcium handling in the patients. The other thing that this slide shows is that there is normal variation between control subjects, right? So that's another thing that we need to kind of tease out at a population level. One thing that we've become very interested in is using control subjects as predictors for disease risk, right? So the two patients I talked about before, it would have been nice to know in advance if they were more sensitive to doxorubicin, in the sense that it was going to give them a cardiomyopathy later. So I think that's one of the projects we have in the lab, is to build a bank of control patients or control subjects and kind of tease out risks for reactive oxygen species damage and things that would make them more sensitive to certain chemotherapy drugs and not others, right? So that's something we're interested in as well. The other nice thing about the iPSCs now is you have a venue to make things work in 3 dimensions, right? So we can now build organoids, and you can use the same biosensors if you want. We can actually see that this is a ball of cells now where the beating is actually coordinated. So this is the calcium biosensor again, and you see it flashes when those cells contract in unison. The thing we like about the organoid, you know, in addition to its effect on making the cells more mature, is that you now start to see multiple cell types there. So we want to kind of look at communication between fibroblasts and the myocytes as part of our work, because we become very interested in what the fibroblasts are doing, right? The myocytes make up 70% of the weight of the heart, but only 30% of the numbers of the heart. So the other cells in the heart are very important, and I think to a large extent understudied, at least. So we came back to this idea that, like, if we have a dish or if we have an organoid with cells on it, how do we make sure we're only studying events in the cells that we want to study those events in? And so we started to build biosensors. We like the biosensors we use because they're driven by very strong promoters, but those promoters are not cell specific. And if you put biosensors under the control of cell specific promoters, you don't get high levels of expression. So what we tried to do then was uh, flox them, right? So put our biosensors so that they're floxed, and then express Cree under the control of a promoter that would be cell type specific, right? So the idea is that you could now only in the cells of interest would you cause the biosensor to be flipped and then expressed under the control of its own very strong promoter. And the idea here is to come back to being able to make sense of what these clusters tell us by only looking at the clusters in the cell type of interest. And this comes back again to the idea that we want to do this in organoids where there are already multiple cell types. In the 2D orientation, we thought about, you know, co-culturing but like we can make 90% of the cells on a dish into ventricular myocytes, but at the end of the day, we want to really examine the communication between the two different types of cells, the fibroblasts and the myocytes, because their relationship evolves during the progression toward failure. So we're also spending some time then trying to develop algorithms that will allow us to parse out these events in the different cells separately, right? So by being able to select on morphological grounds which population of cells is expressing the biosensor, and which population of cells or in those populations can we tease out the individual biosensor responses. So that's kind of something we're also working on. And as I said, it's going to be important when we start to move to the multicellular environment. We bought our CardioExcyte a few years ago, but it's really only a student, Diego Loggia, who's come to my lab in the last year, who's gone nuts with the machine. He loves it, and I must admit it's a pretty cool instrument, right? So we use it in the 2 modes. We use it to measure impedance, and we use it to measure extracellular field potential. And we can see that in like a control subject line and in a patient line, Isoproterenol increases beat rate pretty reliably in both, but the amplitude is really compromised in the patient line, right? Both in terms of the impedance, the extracellular field potential measurements, and when we go back and look at calcium, it seems like there's a strong defect in EC coupling here that reflects a loss of contractility. So we're pretty sure that all of those responses are dose dependent, and Diego is now making everyone in the lab use this machine, and it's becoming quite popular. This again is a very useful tool as a phenotypic screen to track differences, right? So I guess the only one that I need you to look at is this one, the impedance amplitude. So here again is a healthy control, and a second healthy control, doesn't have the same response. So there's variation between patients, but there's marked compromises when you look at the patients compared to the controls, and you can see that, I guess, more strikingly here that the patient samples are very compromised in terms of EC coupling. So we think that this tool will allow us to kind of do a pre-screen for subsequent analysis with the biosensor package later, because it gives us a readout of difference pretty robustly. All right, and so the last thing I wanted to kind of get into was this relationship between the cardiac myocyte and the fibroblast. You can imagine that since myocytes don't they're never replaced, right? So maybe 1% of the myocytes are turned over in the heart over the year. That means the myocytes you have as an adult beat once a second for like 70 years without ever being replaced. So part of that wear and tear is sort of rescued or maintained by fibroblasts, which help turn over the extracellular matrix and keep the cardiac myocytes healthy. But after a heart attack or in sustained hypertension, that relationship alters because the fibroblasts enter a different stage. There is this toggling between quiescent and active during normal wear and tear, but in the face of sustained neural humoral signaling, the myocytes become what are called myofibroblasts, which is an aggressively activated fibroblast, which not just involved in wound healing after a heart attack, for example, but also become a driver of heart failure. So if you think about it, there are things you would like to preserve about the fibrotic response, right? The wound healing aspect, the wear and tear repair aspect, but you'd like to block this myofibroblast state and what it does in driving cardiac myocytes to failure. So in the organoid context, we want to now study that in some detail, but we wanted to also build tools to kind of track the transitions between resting, activated, and myofibroblast state in the fibroblast with a view toward developing some antifibrotic drugs. So we've kind of started to go to using iPSC-derived cardiac myocytes, and now we have ways to kind of make them quiescent. So here it's just DAPI or Hirsch rather, and Vimentin. There's no alpha smooth muscle actin in the quiescent state, but in the myofibroblast state, you can see that alpha smooth muscle actin is massively upregulated. So now we're building a screen which allows us to track how quickly when we release them from quiescence they achieve this stage, and can we impact that with drug candidates. And some of the things we're looking at along that are phenotypic, so gene expression signatures, proteomic signatures, the signaling that we can do, and then some phenotypic things like wound healing and proliferation. And so we can use the same biosensors in the iPSC cardiac fibroblasts. This is just to show you that we can measure ERK signaling in those cells pretty reliably. So we're pretty excited about being able to do that. The reason we want to do that is the reason I kind of discussed a couple of minutes ago, which was that there's two roles for fibroblasts in the heart: the wound healing after a myocardial infarction, and then later the development of heart failure. Certainly, you'd want to preserve the first while blocking the latter. And certainly, we know that there are different sources of fibroblasts in the heart, different types of fibroblasts in the heart. So any strategy that blanket targets all of those fibroblast populations is unlikely to be a viable therapeutic strategy. So what we're after now, and maybe if you invite me back next year, I'll tell you how it's going, is because we have some candidates now that will allow us to shape the fibrotic response, right? So the first job we have then is to understand which pathways we would need to preserve that wound healing, and which pathways we need to block to prevent the cells from going to the full myofibroblast stage, which drives heart failure. So we're very interested now in using the iPSC-derived models to track phenotypic responses under different conditions. And so this is what we're kind of after now with the disease in a dish pipeline. If I had my dream come true, it would be we'd be able to do clinical trials in a dish, and that's like a more of a Joe Wu dream than a small guy in Montreal dream. But we will be able to determine new causes of heart failure. I'm hoping we'll be able to use these systems to better predict risk, and better predict what interventions the patient would need to personalize treatment, predict recovery, predict the ones that will need advanced treatments, and then develop new therapies, right? So that those models are useful for that. All right, so Nanion was very generous to us in the last year in the sense that they lent us an AtlaZ instrument for a few months, and we're just figuring out how to use it now. And one of the things that we're really kind of trying to figure out now is we've become interested in a large family of GPCRs that signal downstream of these lipid mediators called resolvins or lipoxins. Now they're involved in the inflammatory response that also plays a role in the development of heart failure, right? And so resolvins are called that because they actually allow acute inflammation to resolve. But really no one knows how they work. We do know that there's a whole bunch of GPCRs that have these actions in vivo but we don't know what signaling pathways are downstream of those receptors, and we don't know which ligands activate in every case which receptor. And so in some early studies that we didn't do, people have shown that sometimes they can figure out which pathways are downstream of a given receptor, but another group will say that the same pathway doesn't act, or the same receptor combination doesn't signal. So which is it? We don't know often which signaling pathways to examine to understand the in vivo effects. And so in our own hands, the problem is a bit more complicated in the sense that when you look at the lipid mediators, okay, for either inhibition of cyclic AMP or activation of GI, they don't work. But a small molecule drug called BMS gives you a nice robust response, okay, in HEX cells. But that doesn't mean that the LXA4 and the resolvin D1 are not active, because they certainly cause an increase in the production of inflammatory mediators. So maybe we have the wrong cells. That's possible. But we want to know what pathways are operating downstream of these receptors. And that's where the AtlaZ comes in. And we did our first experiment. It was a failure, and it failed for the reason that Ron Knox actually predicted a priori, which was we didn't talk to him before we did the experiment. And after we did the experiment and it didn't work, we talked to him and he explained to us in painstaking detail why the experiment didn't work. So next time we'll have some nice data from that too. But I see great uses for the AtlaZ in several projects in the lab that we have to either characterize large sets of ligands or deorphanize receptors, or try to understand physiological responses in an unbiased way. So we're really happy to have this instrument and hope we can get everything we want done before Nanion comes and collects it from us. Okay, with that, I want to thank the people who've done the work, right? So the core sort of iPSC team is Giada and Diego. These four, Yana, Celine, Ben, and Jaja, they're a part of a small company that we started called Telescope, which is kind of more focusing on the fibroblast. The Heart in a Dish team is shown here, including one of our big donors from the Courtois program. And the permanent staff in my lab, Darlene, Fan, and Domo, I have a picture of Domo. He's been with me so long, with his son on his back as a little guy, and now I have a picture of him with him on his son's back. And then more recently, Robert and Sana are working on aspects of this project, and Deanna is the one testing the AtlaZ instrument in some detail. So with that, I will thank you for inviting me again, and I wanted to thank Nanine again for all of its collegial interactions. I'm happy to answer questions.
Very nice presentation. Thank you very much for taking part. And I hope your wife is happier now that you got all your checks and you showed nice data for it.
She's happier.
Very
Yeah.
good. Are there any questions from the audience today? Maybe I start. You show some differences between your male and female patients for the PK pathways, and they are different in phenotypes. Do you have any thoughts about why that is?
You know, the one thing that we're in the middle of is a pretty big experiment to do like a patient level RNA sequencing and patient level proteomics. We just got funded to do that. I suspect, you know, if it's a loss of pKa tone, it might be a downregulation of aspects of the whole sort of pKa signaling pathway. For the one that's where there's an upregulation of the alpha-1b, it's probably, you know, in the female patient, it's probably simply an upregulation of the receptor, which we will also see when we look at our proteomic data. One thing that I deeply regret doing now is giving up my radioactivity permit, so I can't do binding assays in these cells anymore. But anyway, that's a me problem. But I think that we want to see which, like at a patient level, which networks are altered, and we think that a good starting point for that would be to see which genes are changed with respect to expression in comparing control and patient and controlling patients of different sexes.